Hello, today we're going to be talking about senescence. I'm Delia Orduna.
I'm Shani Henderson.
And I'm Louise Fantone. To start our presentation, I'll be explaining what is senescence. So senescence is part of the reproductive life cycle of the plant. More specifically, it is the final stage of plant development and is characterized by a series of programmed disassembly and degenerative events. In plants, there are two types of senescence, mitotic and postmitotic senescence. Mitotic senescence occurs in shoot apical meristem containing multipotent stem cells, similar to replicative senescence in mammalian cell cultures and yeast. In contrast, postmitotic senescence occurs in organs such as leaves and flowers. Leaves are organs that characterize plants as autotrophic organisms and perhaps the primary source of food on Earth, which use light energy to fix carbon. As leaves age, chloroplast degeneration is initiated, paralleled by catabolism of macromolecules, including nucleic acids, proteins, and lipids. The released nutrients are exported to other developing organs, such as new buds, young leaves, flowers, or seeds, which leads to increased reproductive success. So next, I'll be going over the importance of senescence. To start, 25% of the plant genome is dedicated to genes involved in senescence. Second, senescence is not random, as it is highly regulated and controlled. Lastly, this final stage of plant development generally involves a whole plant senescence process in which all the nutrients from vegetative parts are translocated to the maturing fruit or organ, and the vegetative part dies as a consequence. Vegetative parts include roots, stems, shoot buds, and leaves. They are not directly involved in sexual reproduction, and vegetative parts often are used in asexual forms of reproduction. To the right, you will see a diagram of different kinds of senescence, which from left to right are labeled whole plant senescence, top senescence, deciduous senescence, and progressive senescence. This can help you visualize how they're different from one another. Now I will be going over the different parts of senescence. First, we have proliferative senescence, which is the stopping of cell division. Second, we have programmed senescence, which involves the age-induced, regulated dismantling of postmitotic cells, such as degradation of chloroplasts and other organelles. Last, we have stochastic senescence, which is random degradation of plant cells. For instance, after programmed senescence, the nucleus and vacuoles break down and release contents, the plasma membrane gets holes, and cellular processes get disrupted, which then eventually leads to death.
For leaf senescence, there are two major pathways involved. These pathways are induced by SAG genes in the plant. SAG just stands for senescence-associated genes. The main hormone that is involved in the processes are ethylene, but gibberellins, jasmonic acid, and ABA can also be present and induce leaf senescence. The first pathway is the age development senescence. This is age-induced and involves degradation of chloroplasts and other organelles in the cell. The other pathway is the dark-induced senescence, which also leads to chloroplast degradation. Both of these pathways are also induced by phytochrome and hormones. This is an overview of the age developmental pathway. First, ethylene's presence activates EIN2. Once activated, it then activates NAC transcription factors. I've included on the slides what NAC stands for and what they are. But so after this, NAC then activates ORE1, which is the SAG gene in this pathway. This inactivates GLK1 and GLK2. GLK just stands for golden-like, and these are chloroplast maintainers. So the inactivation of these leads to chloroplast deterioration, thus leading to leaf senescence. Now, ORE1 can lead to leaf senescence by two more pathways. The first one is that ORE1 inhibits BFN1 gene, which leads to degradation of nucleic acids, thus leading to leaf senescence. BFN1 is a gene that helps restore phosphate and nucleotides when a leaf is undergoing senescence, so inhibiting it is needed in this process. The other pathway for this is ORE1 activates SINA1, which is a ubiquitin ligase, and thus leads to protein degradation and then leads to leaf senescence. The other pathway is the dark-induced or stress-induced pathway. This still involves ethylene, and when ethylene is present, it leads to PIF4 upregulation. PIF4 binds to the E-box region of GLK1 and GLK2. It makes transcription come to a halt, and now the plant won't be able to undergo photosynthesis. then this leads to senescence. This pathway, the leaves can be saved if they are exposed to light for more than 2 days and before the 7th day. Once it reaches the 7th day, senescence can no longer be reversed, and it needs to be exposed to light for 2 days in order to grow.
As we know, leaf senescence is associated with aging and is a developmental process critical for plant fitness, which involves genetically controlled cell death and ordered disassembly of macromolecules for reallocating nutrients to juvenile and reproductive organs. These insights suggest that leaf senescence is of substantial biological significance. During senescence, mesophyll cells are dismantled in an organized manner, which changes the cell structure, metabolism, and gene expression. Mitochondria and nucleus stay intact during the end of the leaf senescence process. Chloroplasts are usually the first cells to be dismantled, which allows leaf lipids and proteins to be recycled. This is also why we notice that senescing leaves turn colors, yellow, red, or orange. The remobilization of chloroplast lipids is essential for normal plant growth, the onset of senescence in the future, and the plant's reproductive success. Chloroplasts contain most of the leaf's protein and is a rich source of nitrogen. Nitrogen-containing amino acids accumulate in the senescing leaves. The free and available amino acids provide maintenance of energy, homeostasis, and also supports protein synthesis and nitrogen storage to sink tissues. Nitrogen recycling requires that some organs senesce and die for others to develop. Soil phosphate levels are also usually not sufficient for plants. So during senescence, plants use an elaborate mechanism to remobilize stored phosphate through the degradation of organellar DNA and RNA, as well as the degradation of cytosolic ribosomal RNA. This mechanism also redistributes micronutrients such as zinc and iron from decomposing senescent leaves through the soil and for reuptake to support viable parts of the plant. Senescence is important for seed viability because when we decrease phosphate remobilization, we reduce the germination rates and the total phosphate levels in the seeds. This return of macro and micronutrients to the circulation of the system is necessary for nutrient storage and plant survival. Senescence is important during environmental stress because plants can use this process of senescence to promote survival and reproduction during the times when the plant is exposed to extreme stressors. This allows plants to respond to its fluctuating environment and could be beneficial for agriculture because leaf senescence is of critical importance for improving crop yield. Understanding the regulatory mechanisms of leaf senescence will provide valuable clues and a theoretical basis for manipulation of this trait in agronomically important plants. So when senescing leaves fall to the ground, they enrich the soil with nutrients, which aids seed germination and plant growth. Senescence promotes the efficiency of plants by shedding older organs in order to recycle nutrients for new growing parts of the plant, such as new tissues, fruits, flowers, and other reproductive organs. The eventual complete shedding of all leaves is helpful in reducing the plant's transpiration as well, which enhances survival induced by extreme environmental and nutritional conditions such as biotic and abiotic stresses, darkness, phytohormones, and oxidants. Thanks for watching. And these are our sources.